Hi, my name is Robin Ayala. Today's date is January 4th, 2020, and I'm here to discuss the role of the pancreas and liver in the regulation of digestion. Okay, so we will be discussing the pancreas and the liver. Both two are necessary for stomach content digestion. The pancreas secretes digestive enzymes and sodium bicarbonate. The liver secretes bile. The pancreas has two types of secretory epithelium. You can see here the pancreas to the right, and here is a better view, microscopic view of the two secretory epitheliums. The endocrine islet cells, right over here, secrete insulin and glucagon to control the sugar levels in the body. And then exocrine Acini lobules, which secrete digestive enzymes and bicarbonate, and that is seen over here to the right. So the Acini lobules secrete zymogens, which are inactive enzymes and must be activated by trypsin to activate the cascade that you see to the left. So zymogens and trypsinogen are what initially are secreted by the pancreas. Trypsinogen needs to turn into trypsin by the enteropeptidase in the brush border, which activates trypsinogen to turn into trypsin, which then activates to the left the zymogens to turn into activated enzymes, which is like chymotrypsin, carboxypeptidase, the colipase, and the phospholipase, all necessary for digestion. The signal for release, distension in the small intestine, so like food causing you to stretch, neural signals, and the CCK hormone. Pancreas also secretes bicarbonate, which is essential to neutralize acids in the stomach because stomach contents have an acidity level of 2 usually. So with the more neutral acid level, digestive breakdown is more effective. It is secreted by the pancreas, mainly a little bit by duodenal cells, but mainly by the pancreas, and requires high level of carbonic anhydrase to create this watery fluid to neutralize the acids. You can see here in the picture to the right, the chloride enters the cells by indirect active transport and leaves the apical side through a CFTR channel. Chloride then re-enters the cell in exchange for bicarbonate. The leaky junctions allow the paracellular movement of ions in water. Negative ions in the lumen attract sodium by the paracellular pathway, and water follows. Oopsies. Go back here. The liver secretes bile, which is also very important. It's secreted by the liver cells or hepatocytes, and then stored in the gallbladder, right here, it's green, and goes down to the common bile duct. These bile contains bile salts for enzymatic fat digestion, bile pigments, which are like bilirubin for waste products of hemoglobin degradation, cholesterol, drugs, and other xenobiotics. So bile salts are essential to break down large fat droplets to be digested. But they are not altered during fat digestion. They are reabsorbed back into circulation in the ileum, and if they make it to the colon, they're converted back to bile acids and return to liver to be resecreted. Every time we eat, they are cycled 2 to 5 times for each meal. So it's a little bit of recycling going on. And you can see here the liver, how how it is absorbed from the GI tract. So bili, nutrients, drug, foreign substances. It goes to the hepatic portal vein, and then in the liver, they have glucose, fat metabolism, protein synthesis, hormone synthesis, urea production, detoxification, and storage. And then leaving the liver to the bile duct, they're secreted into the duodenum, which are bile salts, bilirubin, and water, ion and phospholipids. And then over here to the right, metabolites and drugs from peripheral tissues go from the hepatocyte to the liver. Bilirubin, nutrients, metabolites. And then from the liver, it goes through the hepatic vein. Glucose, plasma proteins, albumin, clotting factors, urea, vitamin D, and metabolites. So liver is essential for digestion, and no more than digestion is filtering of other things in the blood and either absorbing or getting rid of stuff through the circulation. And that is the end of the presentation. Thank you so much.